second year. It's Dr. Yasin. आज का सर्जिकल लेक्चर जो है is going to be about tissue engineering. ठीक है. I'm sure a lot of you already have a rough idea of what tissue engineering stands for, what could be its possible uses. You might have already heard of it. Just from prosthetic valves to artificial limbs, all come under biomedical engineering and tissue engineering, basically. So, फिर आज के lecture में जो है we're gonna talk about tissue engineering, ठीक है, and उसके relatively terminologies, उसके साथ-साथ different examples and everything as well. So, yeah, let's start today's lecture. Bismillahirrahmanirrahim. Tissue engineering, ठीक है, or also known as regenerative medicine, because we'll discuss it briefly. If you're looking on the left side, we have a picture. इसमें जो है they're showing a tissue engineered bladder on the left side, along with a trachea, which has been seeded with stem cells, where they took stem cells from possibly another trachea and they grew it into a functioning large size, normal size trachea, as seen on the right side. So we'll be talking about introductory level of tissue engineering basically in this slide. What are stem cells, tissue model constructs, bioreactors, lab techniques that are used or gone into tissue engineering, and along with that, types of signals that are used, growth signals specifically, and ethics as usual involved in everything. Definition of tissue engineering. There's a quite famous professor, Professor Robert Langer, and his say in this matter matters because he's one of the leading entrepreneurs in the field of tissue engineering or biomedical engineering. And he's currently, I believe, still a professor at MIT and Harvard. According to him, tissue engineering is a field which applies principles of biology and engineering to the development of functional substitutes for damaged tissues and whole organs. It's a sort of replacement or substitution of damaged tissues and whole organs that we construct using our biomedical engineering knowledge. Why do we need them, and what is the need for tissue engineering? Let's give multiple applications for tissue engineering. In regenerative therapies, they also have proved to be a potential. And tissue replacement for congenital defects. Just to know, we have a classic example of cleft lip and cleft palate, which are congenital defects. In disease, just on liver, kidneys, diseased organs, unमें जो है transplant करना, that's another potential for tissue engineering. And then trauma, battlefield wounds as well. What does it do? It improves the performance of that limb, organ, whatever it is that we are engineering. Models to test the therapeutic efficacy and also toxicity. Again, these are just some of the examples of tissue engineering, how advanced it's gotten, and what they've been able to achieve in labs. The topmost picture just now, we saw it earlier as well, as the bladder, trachea, multiple other things as well. They've been able to, even a year, gone big. Now, how or what goes into the process of tissue engineering is important as well. So the process of tissue engineering starts initially with cells that are isolated from the patient. Secondly, these cells are cultivated until a cell bank is created. Then different biomaterials are utilized in order to fabricate scaffolds. Now, the next step after that is to seed the scaffold with the cells. It's basically growing them. Now, finally, the engineered scaffolds, along with the cells, are placed in a bioreactor, a device which mimics the conditions inside the body, which helps the growth of the cells. Cells proliferate in an environment that is suitable to them, which is most likely mimicking a condition of inside the body. Now, these seeded scaffolds can be bathed with growth factors, enhancing their further development, and with be mechanically altered as well. This is sort of high knowledge, but in order to stimulate cellular growth and proliferation, bioactive environment. यहाँ पे diagram है, we see there's the patient from which the cells are isolated, we biopsy, and then along with that cells को हमने further isolate कर दिया. उसके बाद cell cultivation हुई, scaffolding में डाला, cell proliferation होगी, hit it with growth factors and mechanical stimulus, and then we're back at where we started, implantation into the patient. Now, tools that are required for tissue engineering. क्या क्या? Obviously cells, that is the necessity, absolute necessity, which is a living part of the tissue. Then what do we need is scaffolds. These provide structural support and shape to construct, provides place for cell attachment and growth. And along with that, we need bioreactors and cell signaling. These are signals that tell the cell what to do and enhances this routine and mechanical stimulation, and provide a basic environment in which it can do so. So we have the cells, scaffolds, and then biological factors that are main components of the tools for tissue engineering. Step 1, acquiring the building blocks. इसमें जो है cell sources. Cell sources क्या हो सकते हैं? There are multiple types, like we're going to discuss right now. Autologous, meaning they come from the person that needs the new cells. उसी बंदे से जो निकालेंगे cells, that's known as autologous. They have the fewest problems, obviously, जिस बंदे से निकाले उसी में cells inject करेंगे, so there's less chances of pathogen transmission, मतलब diseases वगैरह जो है, automatically chances of spreading those is eliminated, or even reactions due to difference in pathogens or cell receptors जो है वो भी नहीं होगा. However, in some cases might not be available. That's autologous. You cells उसी बंदे से निकाल के and you inject it into the same person. Allogenic means come from a body from the same species. Look, obviously, if you're going to inject it into humans, but from a different person. You take it from one person and you inject it into the other person. That's allogenic, same species. If you take it from animals and you inject it into the animals, that's allogenic for animals. Xenogenic come from a different species than the organism they're going into. Obviously, from animals to humans to animals, that'll be xenogenic. Isogenic, sink cells are isolated from genetically identical organisms such as twin clones or highly inbred research animal models. That's isogenic. Genetically completely identical, such as twins, clones, highly inbred. Now, extractions from fluid tissue such as blood cells are extracted by bulk methods, usually centrifugation or apheresis. Now, from solid tissues, it can be done as well. Extraction is more difficult though. Usually the tissue is minced and then digested with enzymes such as trypsin or collagenase, उससे जो है extracellular matrix remove कर देते हैं, that holds the cell. उसके बाद cells are free floating and then extracted using centrifugation or apheresis. Stem cells. What are stem cells? They are the undifferentiated cells that can have the capability of self-renewal and have the potential to differentiate along one or two lineages. Over here, like we see stem cells in this diagram. Culture stem cells can become any cell in the body, so they have the ability to produce nerve cells, blood cells, liver cells, even muscle cells. And they can self-renew themselves as well. The totipotent means can produce all cell types. Pluripotent can produce most cell types. Multipotent can produce more than one cell type, or they could be of unipotency, can produce only one cell type. This will be totipotent stem cell that can produce all cell types. Will be now types of stem cells. There's multipotent stem cells. It's showing you right here. Now stem cells found in adult brain, blood, cornea, retina, heart, fat, skin, dental pulp, bone marrow, blood vessel, even muscles are hematopoietic stem cells. Mesenchymal stem cells or stromal cells. Endothelial progenitor cells. Neural stem cells. They're the different types. Then you have the embryonic stem cells as well. Human embryonic stem cells. They're cultured cell lines derived from the inner cell mass of the blastocyst that can be grown indefinitely in their undifferentiated state, yet also capable of differentiating into all cells of the adult body. So they're derived from 5 to 7 day old blastocysts that are precursors to embryos, and they are pluripotent in nature and capable of generating any cell in the body, any kind of tissue, basically. That's the definition of embryonic stem cells. Okay. Now, then we have the fetal stem cells. To get these stem cells can be obtained from the blood, bone marrow, and other tissue of aborted fetuses, fetal stem cells. They proliferate in vitro as efficiently as embryonic stem cells and are pluripotent as well. Now, induced pluripotent stem cells. What is this? It's these specialized adult cells, right, that could be reprogrammed using genetic manipulation to become embryonic-like induced pluripotent stem cells. Okay, how do they do this? Is they use retroviral or lentiviral transfections to get out. Retroviral vectors do not insert their own genes into host cell genomes to reduce the risk of tumor genesis. Synthetic RNA combinations of small molecules, growth factors, they act as transcription factors and obviate the need for viral factors altogether. Now, the major features of different stem cells include the types used in tissue engineering most commonly. We have the different cell types in this table, lined on the left side. The ease of availability, its expansion in vitro, its pluripotency, ethical concerns that might be involved in risk of malignancy, autologous, and whether it's likely future use, the outlook. Okay, so say ease of availability, it's limited to get for somatic cells, and it's moderate for even embryonic stem cells, even fetal cells, but it's good for ESCs. So again, these are basic cell types and their efficacy in usage in tissue engineering. There are different factors, like we highlighted, and that determines its efficacy in the use of tissue engineering, basically. So that was all about the stem cells. Taking the other component or the tools that are needed in tissue engineering were the scaffolds, right? The materials and their production. So what this guy is showing is the structural component that has been made to be artificially induced, and on which the stem cells can be implanted and growth can be promoted. Scaffolds could be three-dimensional materials constructed in order to provide structural and developmental support for the tissues to allow the cells to adhere to it, proliferate, differentiate, and most importantly, secrete extracellular matrix. All right, so they're showing the scaffold in this picture, and then they've zoomed in on it microscopically and shown you the integrity of each material involved in making that scaffold. Scaffold construction requirements. Scaffolds should provide void volume for vascularization, new tissue formation and remodeling. 2, it should have high porosity, porous, so have suitable pore sizes, and the pores should be interconnected. 3, biomaterials should be degradable upon implantation at a rate matching that of the new matrix production by the developing tissue. 4, the biomaterials used to fabricate the scaffolds need to be compatible with the cellular components of the engineered tissues and endogenous cells in host tissue. And 5, scaffolds provide mechanical and shape stability to the tissue defect. These are the construction requirements of the scaffold. Okay, it should be degradable, compatible, mechanically support the stability, highly porous, and void volume for vascularization, further tissue formation, and then remodeling as well. All right, we have further examples of the scaffolds that provide mechanical and shape stability to the tissue defects. This is sort of a scaffold that can be applied superficially and adheres to the skin and promotes the healing. Now, biomaterials for scaffold. Materials that can be used for scaffold. It could be natural biomaterials or synthetic, mainly. Two main differentiation here. About natural biomaterials, maybe we have protein origin biomaterials or polysaccharides origin biomaterials. Under protein, we have silk, collagen, fibrin, gelatin. Polysaccharides, maybe we have hyaluron, alginate, agarose, and chitosan. These are all different materials. You don't need to know this in depth. However, these are the natural biomaterials. You get that commonly used, can be used for biomaterials for scaffolding. The other ones are the synthetic biomaterials. It's polymer based, yes, ceramic polymers, where we have a whole lot along with even ceramics as well that can be used. Let's see, what are some of the methods used for synthesis of tissue engineering scaffolds? For synthesis, we can use nanofiber self-assembly, textile technologies, solvent casting and particulate leaching, gas foaming materials, emulsification, freeze drying, thermally induced phase separation tips, electrospinning is another method, CAD, CM technologies, laser assisting bioprinting. Bioprinters or 3D printers are also another form of leading methods used in synthesis of tissue engineering currently. CAD-CAM is basically another relative term for 3D printing. CAD-CAM technologies. This is called a ghost organ, ghost. It has no cells to it. It's only structurally built by connective tissues. Now the patient's own cells will basically replace those will take place inside this connective tissue because it's a ghost organ. It's already belly cells in here. To get the patient cells in which this organ is transplanted will reseed and regenerate the organ and then eliminate the tissue rejection in organ transplants. So that's one way. By injecting ghost organs, you eliminate the possibility of tissue rejections, or that is commonly a case in transplants. Then there's natural scaffolds as well. Laser assisted near hearts are basically examples, and these are laser assisted bioprinting. This is another example. They're basically, you can see they're printing the organs level by level, basically they're building up onto it. Clumps of cells are smidge of organs can be built up there by printing clumps of cells onto gel that turns solid when warm. Which is like glass slide, उसके ऊपर thermal reversible gel रखी, a clump of cells are being injected by a modified inkjet printer. But once the cells have fused, the gel can be removed simply by cooling it. Obviously, you can all make out basically what tissue you're going to have. This is part of the face. This man, lower part of the nose involved, the lips, cheeks, and chin. And in this, you can see it's the ear. Okay, the third important step, bioreactors. I'm sorry. Bioreactor. It's a tissue engineering bioreactor that can be defined as a device that uses the mechanical means to influence the biological processes that is needed. Take a bioreactors, measure tissue culture may be done on petri dishes. Take a simple method commonly used in tissue cultures. Now, bioreactors stimulate the body conditions. and there's different things that it can simulate, sorry, like the gas, the temperature, pH, and the amount of oxygen. Okay. Now, it's basically bioreactors designed to perform at least one of the following 5 functions. क्या क्या इसमें? A, keep facilitating mass transport of the tissue. Maintaining desired concentration of gases and nutrients in culture medium that is going to further promote its growth. Third, provide a spatially uniform cell distribution. Fourth, expose the construct of physical stimuli. Along with that, provide information about the formation of 3D tissue. So it's referring to any manufactured device or system that supports the biologically active environment. And इसके साथ there's chemical processes that are being carried out, which involves organisms or biochemical active substances, which could be derived from such organisms. और ये process जो है aerobic या anaerobic भी हो सकता है, ठीक है? And they're usually जिस तरह तस्वीर में देखी है, just shown a picture right here of a bioreactor, ठीक है? Cylindrically shaped जो है, इसके मुख्तलिफ sizes भी आते हैं from liters to cubic meters and are often made of stainless steel, as it can be seen in this picture as well. So its main function is to, or the way it's designed is to grow the cells, your tissues, in the context of the cell culture. And they're being developed for use in obviously the tissue engineering purposes. There's this different type of bioreactor as well, which is known as the spinner flask bioreactor. In this, we can see there's a flask that's right here. There are multiple scaffoldings inside it. Cell suspensions are on the scaffolding. A magnetic bar in the bottom, obviously for the spinning purposes. Here's another picture of a hydrostatic pressure bioreactor. These are all just different types of them. There's like photobioreactors as well that are there. And then there's bioreactors for specialized tissues as well, like PCM. This is a flow perfusion bioreactor, because 3D perfusion bioreactor is quite complicated, as you can see. A lot of things go into manufacturing one of these. Now, okay, bioactive environment, the fourth part. The bioactive environment in tissues engineering can be created with the utilization of 3 main things: growth factors, hormones, and then cytokines. These are your 3 main components in promoting a bioactive environment for tissue engineering. Initially, इसमें भी वही step है, we have isolation of cells from body. As can be seen right here. Sorry about that. Isolation of cells from body, seen right here. उसके बाद जो है cells undergo expansion process. And then we have seeding into appropriate scaffolds. Injectable scaffolds and then porous scaffolds. उसके बाद we place them into the culture to promote its growth. And then they're finally injected directly back into the patient or transplantation by surgery. What are some of the considerations that should be gone into it? There's pros. Obviously, everything is pros and cons. The pros for this is that it can help a person change or conquer his disease or illness, and that can change his views towards himself and society and everything. The person will go through fewer surgeries. Fewer surgeries, better outcome. No chance of rejections in this. People would not have to wait for an organ donor. There are people that are on waiting list for months or even years at end to find a perfect match with a donor, and then once they do find it, it's quite expensive. Those it can set them back economically as well. People would not have to donate their organs ठीक है after they die. तो एक वो भी जो है इसलिए वो भी हल हो जाता है. This technology could lead to even greater technologies in the future, and then it's a permanent solution. It's not something that's temporary. However, okay, on the other hand, it takes a lot of research and understanding of each organ and tissues. Medicine researchers face many difficulties in constructing suitable scaffolds. It's not that simple to create scaffolds. And the researchers, and they require a lot of funding as well. So उसकी वजह से financial issues ठीक है जो लोगों के अपने in order to afford that, because the researchers involve a lot of money, so obviously this is a very costly field, right? And then there are obviously ethical issues as well, where people don't feel comfortable going through with this, and we'll look at this ethics wise as well. No, actually, this is it. So if you guys have any questions about this, please feel free. However, I just wanted to go over the other types of bioreactors that we can see as well. In this, basically, we discussed a few. Yeah, taking the bioreactors की जो हम बात कर रहे थे, जिस तरह इसकी तस्वीर भी दिखाई थी, ये एक किस्म का जो है, ठीक है. This is the general picture of bioreactors that is commonly seen. ठीक है. बाकी जो होते हैं, there's a photoreactor, ठीक है. A photoreactor bioreactor, which incorporates a light source, ठीक है. It could be natural sunlight or artificial, ठीक है. And इसकी वजह से virtually any translucent container could be called a photobioreactor. However, the term is more commonly used to define a closed system as opposed to an open storage tank or pod. ठीक है. बाकी एक होता है bioreactor for specialized tissues, ठीक है. उसमें many cells and tissues, especially the mammalian ones, must have a surface or other structural support in order to grow. And agitated environments are often destructive to these cell types and tissues as well. So there's a lot of engineering that's involved in this field as well. And tissue engineering is basically biomedical engineering. It's an up and coming field, which is still being researched, and definitely has a lot of potential. So we still have obviously a lot of time. So what we're going to do is basically roughly just go over it once more, just to have a quick review of the main points. You may not get your points, and we'll just glide over them. So tissue engineering का हमने उसकी applications की बात की, ठीक है, where they can be used, ठीक है, for regenerative therapies obviously, लेकिन where they can be used, they can use amongst multiple congenital defects, like we said, basically for इसमें जिस तरह show किया हुआ in this picture right here, cleft lip and cleft palates, ठीक है. Other places they can be used to replace portions or whole tissues. हमने बात की disease to transplantations without liver, it could be bladder, even kidneys, liver, bladder, bone, cartilage, blood vessels being इस्तेमाल कर सकते हैं even. Most superficial thing, which is skin, ठीक है. इससे क्या होता है? It improves the performance, ठीक है, and the models to test therapeutic efficacy and toxicity as well currently. All right. Need क्या है इसकी tissue engineering की? Tissue engineering holds the promise of producing better organs for transplant. हमारा goal जो है कि tissue engineering is in vitro construction of transplantable vital tissues. ठीक है, and they can correct many incurable genetic defects like we discussed earlier. The supply of hard and soft connective tissue, and along with other applications of this as well. All right. Over here, we discussed it shows a nice picture of multiple tissue engineering techniques that are being used in transplanting or creating multiple organs. And we have a bladder, trachea, skin, ear, ठीक है. Obviously, हमने tried की बात की थी, three things that are the tools that are necessary, right, for basically tissue engineering: the cells, the scaffold, and the signal molecules. ठीक है. So obviously signal molecules they क्या growth factors, hormones जो involved होती हैं, ठीक है ना, and then proteins as well. I believe we looked at this slide, right? In this slide, it's highlighting all the tissues, take examples of tissues that are created by tissue engineering, okay, and then how they can be used to treat skin, obviously, genetic conditions of the burns and skin defects, cardiac muscle, okay, जो patients that have suffered from heart failure, yeah, more specifically heart valves. They've created tissues constituents of heart valves, okay, in South1 conditions. Where congenitally acquired valvular heart disease, cartilage, degenerative and traumatic joint disorders, trachea, bronchus. We saw a picture of this earlier. So, congenital acquired stenosis हो गई हो, resection for malignancy, ठीक है ना? Bladder, जिनमें congenital bladder malformation हो या cystectomy जिनकी हो गई, again due to malignancy or other problems. And anal या bladder, again sphincter incontinence की वजह से, pancreatic islet cells जिनकी insulin dependent diabetes होती है, ठीक है, उनमें could be beneficial. Large blood vessels, atheromatus, ठीक है, जिनमें atheromas मतलब aneurysmal हो या traumatic arterial disease, ठीक है, laceration हो गई, everything. Esophagus में just a benign stenosis and resection, way for malignancy again. Yes, में congenital भी होते हैं, benign stenosis, congenital atresia and all. This may be esophagus tissues could be helpful that are tissue engineered. Yes, small intestinal failure, or surgical resection for cancer, or ischemia. See, these are the different tissues, examples of tissues that are being created by tissue engineering, and they're helpful or they're benefiting certain conditions. Okay, moving on. We said how this is done. Obviously, process come and described together as seen in this picture. Basically, starting from a biopsy where the cells are taken from the patient, the cells are further isolated, then they are cultivated until a cell bank is created. Self-cultivation or cell bank is created, proliferation happens. Then these different biomaterials are utilized in order to fabricate these scaffolds, which is a part of the triad for tissue engineering: cells, scaffold, and then comes the signaling molecules, the mechanical stimulus, growth factors, proteins that are used, and the bioreactors, obviously, in which the engineering scaffolds are placed. So there's also strategies that can be used in engineering tissues. They can broadly be classified in three major classes. Before going into these tools, उसमें conductive, inductive, and cell transplantation approaches की होती हैं. And obviously, just हम बात करें techniques, जो methods हैं that can be used is in vitro and in vivo. What does it mean? Basically, in vitro, जो है construction in lab, of the vital tissue, और उसकी subsequent implantation post body में. इसकी advantage is ability to examine tissues as they are formed. So you have constant observation on the tissue that is being grown in the lab, and to perform specific tissue measurements as well along with it. By in vitro tissue engineering of tissues such as bone, need for recruitment of specific cells to site is negotiated, and predictability of regeneration is enhanced. This overcomes many of limitations with conventional therapies. However, disadvantage is absence of a physiological environment that is suitable for the tissue being engineered. And implanted tissue has to be incorporated with the surrounding bone. In vivo indicates that obvious advantage of tissue regeneration in vivo, in which incorporation occurs as tissues are formed. And this has formed basis for tissue engineering, which now includes implantation of porous matrices, जिसकी हमने बात की थी कि scaffolding की requirements में कि porous matrices seeded with appropriate cells and signaling molecules to facilitate tissue regeneration in vivo. All right. उसकी requirements में सारी थी. Disadvantage के in vivo approach में कि regenerating tissues may get dislodged or degraded by mechanical forces acting normally inside before regenerated tissue is fully formed and then incorporated. ठीक है, तो cells की हमने बात की, scaffolds की भी बात की, bioreactors and cell signaling. ठीक है, the main components of the tissue engineering. And then we talked about how to acquire the cells, which are the building blocks. Autologous, coming from the same person that needs the cells or needs the transplant. We use those cells to grow the tissue. Allogenic, coming from same species. Xenogenic, coming from different species. Isogenic, isolated genetically from an identical organism, such as twins, clones, hybrid, inbred research animal models. Extraction की हमने बात की थी from fluid tissues and then stem cells भी हम बात थे, then stem cells की हमने बात की थी कि they're the undifferentiated cells that can self-renew and have the potential to differentiate along one or two lineages. And we talk about their efficacy could be pluripotent, can produce all cell types, pluripotent, can produce most, multipotent, can produce more than one, unipotent, can produce only one. Number of criteria should be satisfied in order to achieve effective, long-lasting repair of damaged tissues. इसमें A, adequate number of cells should be produced to fill the defect. दूसरा कि cells must be able to differentiate into desired phenotypes. Okay. And these cells must adopt appropriate three-dimensional structural support. They must be able to appropriately adopt the structural support with scaffold and produce the extracellular matrix. Fourth, will have produced cells must be structurally and mechanically compliant with the native cell, the initial cell. 5, cells must successfully be able to integrate with native cells and overcome the risk of immunological rejection. 6, there should be minimal associated biological risks involved in this as well. Okay. Further, we talked about the types of stem cells, and then obviously looked at specifically the embryonic stem cells as well. But when we're trying to supply these exogenous cells into defect, we're looking at two modes, which are cell seeding, cell suspension. Cell incorporation into the implantable matrices should be ensured that localization at treatment site. Concept being referred to as cell seeding. Alternative is to inject a cell suspension into a sealed compartment containing the defect. Some cells की characteristic features they're capable of dividing and renewing themselves in their body for long periods, and they are unspecialized. They can give rise to specialized cell cells. Stem cells have the ability to renew. Now, they could be adult stem cells, embryonic stem cells. main broad classifications. The adult stem cells they're known as somatic, from the Greek definition body, somatic. And they have stem cells in germline, giving rise to gametes. Stem cells can be found in children as well as adults. ऐसा नहीं कि नाम adult stem cells तो won't be found in adults, but no, they're found in children and adult stem cells. Pluripotent adult stem cells are rare, and they're generally small in number, but can be found in a number of tissues, including umbilical cord, umbilical cord blood, sorry. And most adult stem cells are lineage restricted, and they don't have multiple lineages that they could transform into, and are generally referred to by their tissue origin, just like embryonic stem cells. Okay. Now, embryonic stem cell lines, they're cultures of cells derived from epiblast tissue of inner cell mass of a blastocyst. Okay. And or blastocyst, earlier morula stage. And approximately 4 to 5 days old in humans and consisting of 50 to 150 cells. Embryonic stem cells are pluripotent and give rise during development to all derivatives of 3 primary germ cells. क्या होती हैं 3 primary germ cells? Ectoderm, endoderm, mesoderm. That arise from the embryonic stem cell. Broadly, जो हमने वो देखा था, this is important. Pluripotent, each cell can develop into a new individual. Pluripotent cells can form cell types. Multipotent cells differentiate but can form a number of other types. Oligo, ability into few cells. Uni, ability to produce cells into their own type, self-renewed. This is adult muscle stem cells. They are the example of unipotent stem cells. All right. See, step 2, जो हमने बात की scaffolds की, and the materials that go into its production. Obviously, scaffolds, the main purpose क्या है कि guide, organize करना for tissue engineering. It helps with the growth and differentiation of the cells in process of forming functional tissues. They provide, obviously, just a structural element, the physical and the chemical signals that are necessary as well. Tissues, they're composed of cells, insoluble extracellular matrix, ECM, and soluble molecules to regulate cell function as well. Obviously, we discussed. This is just the enhanced picture, some of the three-dimensional view. Construction requirements can be बात की थी कि wide volume, highly porous, degradable material, compatibility, and provide mechanical and shape stability as well. Types of scaffolds हमने बात की थी, whether it could be broadly categorized into absorbable, non-absorbable, right here. Natural, synthetic, वही हो गया absorbable या non-absorbable. Synthetic is non-absorbable, natural, absorbable. Sorry, my bad. Yeah. Synthetic ceramics can be used as well. These are implemented as matrix materials for facilitating regeneration in vivo. And most widely used forms are the triclinium phosphate, hydroxyapatite as well. Okay, इसी में जिस तरह natural जो है biomaterials that are used, these are the absorbable, like you said, and इनमें जो collagen जो है, it's a protein, right? I'm sure you've all heard of it. It has a 3 polypeptide chain. ठीक है, इसमें collagen जो है constitutes almost about 1/3 of all protein in the body, and accounts for almost 60% of gingival connective tissue. So you get 90% of total protein is present in the bone, collagen. Okay, it's medical devices derived from animal sources, bovine skin, tendon intestine, or sheep intestine as well. Collagen-based sutures, hemostatic sponges have also been used. resorbable collagen barriers have been used clinically for certain procedures. ठीक है. Although their combination with biological modifiers has not been explored yet. ठीक. Right. So ideally, scaffolds should be three-dimensional, just that we discussed earlier. And as you can see in these pictures as well, of all the scaffolds that we've seen, including this. Ideally, they should be three-dimensional, cross-linked. Into pattern joints, as you can see, they're crossing the fibers inside it. The material that's been used has been cross-linked, porous, like we said, biodegradable. Void volume, for vascularization, and provide mechanical stability as well. उसके अलावा biocompatible हो, natural healing, so signaling molecules के ऊपर the third most important part. There are the biological modifiers, and their materials or proteins and different factors that have potential to alter the key cellular events in the host tissue by stimulating or regulating the wound healing process. Obviously, इसमें जो है different types of signaling molecules that come involved. Systemic हो सकता है या local. Systemic endocrine आ जाता है PTH, parathyroid hormone, growth hormone, yeah, local as well. And what they do is basically further enhance the growth of the tissue. इनकी लेकिन classification हो सकती है कि एक जो है either they promote growth and differentiation factors. Two, either extracellular matrix proteins produce those. Yeah, third, mediators of the bone metabolism. Growth factors में जो है we get we have all of factors such as platelet-derived growth factors, insulin-like growth factors, transforming growth factors, fibroblast growth factors, and bone morphogenic proteins as well. These are under the category of growth factors, and they play an important role in regeneration. If you were to take basically platelet-derived growth factor, if you're looking into platelet-derived growth factor, basically it's 3-isosomal PDGF, and they have unique binding properties. Then if we're talking about mediators of bone metabolism, we have several agents as well which affect the growth of bone. Some prostaglandins, glucocorticoids, and bisphosphonates. Platelet-rich plasma होनी चाहिए. अच्छा. We discussed the pros and cons, but what could be some of the limitations of tissue engineering therapy? This is important. You should know this as well. Pre-culture condition of donor cell. Survival of transplant. Local immune response to cellular grafting. Pre-culture condition of donor cell. Human bone marrow stromal cell lose their in vivo osteogenesis ability during in vitro expansion. The fate, the survival of transplanted cell is obviously uncertain, but cells die quickly or migrate out of transplanted site, and cells do not survive usually more than 3 weeks. इसलिए जो है has to be closely monitored and regulated, and that's why we have the bioreactors which provide just a uniform cell distribution. They maintain the desired concentration of gases and nutrients that is required by the tissue in the cultured medium. They also facilitate mass transport of the tissue, expose the construct to physical stimuli, and provide the information about the formation of 3D tissue. So we talked about then the different types of bioreactors. Okay, we looked at that as well. Uh, then we have this nice diagram which kind of highlights the whole process through which tissue engineering goes through, the isolations of the cells, to get expansion of the cells that, and then along with that, seeding into appropriate scaffolds that are created, the porous, injectable, different types. Injectables are placed into culture and then further directly injected back into the host. Yeah, surgically transplanted back to the host. So again, then we talked about the few considerations that should be taken as well. The pros, obviously, that it can help a person conquer disease or illnesses. There's but less chance or no chance of rejection. People would not have to donate their organs after they die. Permanent solution, TICA technology can lead to other findings and further enhancement in our technologies as well. TICA, obviously, along with pros, there are cons. However, there are fewer at the moment, but they're of higher weightage. Financial issues, main concern. TICA takes a lot of research and time in understanding each organ and tissues and their engineering methods. TICA, so I understand this is quite difficult of a topic, tissue engineering, but you just need to know the basics like we went over. Uh, that's more or less important for you at this moment. Um, and the possible applications of tissue engineering will be zuri janna kanka, where different certain diseases or in conditions where you can use tissue engineering. See, you guys have any questions?